I call this chapter Beyond Mendel because Mendel was just working with genes that had two traits and that they were one dominant, one recessive, but there's a whole lot more going on in here, okay? So that's why I call this Beyond Mendel. This is still genetics, but it's stuff that was discovered after Mendel, okay? Incomplete dominance. Sometimes you don't have a dominant and a recessive, but it seems that the phenotypes blend, okay? The phenotypes blend in there. So for instance, you have a red flower, big R. You cross it with a white flower, that's little r. By Mendel's idea, the big R should be red, but when you have incomplete dominance, you get a color that's between red and white. So you get pink, okay? That's very common. In fact, when I was trying to replicate Mendel's experiment with flowers, I had a time finding a flower I could plant in the yard that would grow quickly and that is not having complete dominance. Most of the yard plants showing complete dominance, and it took me a while to find the right flower. You could have codominance. What if both genes are dominant? In codominance, both traits are expressed. So you're crossing a red flower and a white flower, you don't get red, you don't get pink. You get a flower that has some parts red and some parts white because it expresses both of them. And that actually happens. I put a picture in there in case you thought that I was making that up. By Mendel's genetics, it should be flower. By incomplete dominance, it should be pink. You don't have pink in there. You have red and white. So that's codominance, meaning together. They're equally dominant. And this is something that tends to cause problems with the students, the difference between incomplete and codominance. Okay? The blood groups in humans, and this is not all the blood groups, but this is an example of codominance because you have an A allele and a B allele that are codominant. Okay? So when both alleles are A, you form an A antigen and you have A group. When both of them are B, you express the B antigen, you have B. When both of them are expressed because you have A and B, you express both A and B on the cell and you have the AB group because they are co-dominant. Don't worry now about the little I because we are going to get to that, okay? But the reason you have group AB is because A and B are codominant, so you express both. Now, multiple alleles, this is less common, but sometimes you have more than two alleles, which increases the number of combinations. Once again, we go back to the blood. Yes, we have the dominant A, IA, the dominant B, but there's also a recessive. This one does not produce anything. So basically, big IA or big IA, the recessive, both of them are going to produce the A antigen, produce A. This is what we're seeing here. But what if you have the homozygous recessive? It doesn't produce any antigens in here, so that explains group O. So IA and IB, and notice the I is capitalized, they're dominant, but they're codominant. So that explains groups A, B, and AB. The little I is recessive. And when you have the homozygous recessive, you don't have any problems being expressed, so you have group O. Group types are going to be a combination of multiple alleles and codominance. Multiple alleles explains O, codominance explains group AB. Sometimes it's not multiple alleles, but multiple genes. And we call that polygenic, meaning many genes. Okay? And you find that looking at the eye color. Look how many different eye colors. To go from the pure blue to the pure brown, you have all of this combination where you have more blue and a little bit of brown, and the brown increases and you get to pure brown eyes. Same thing with the skin color. You could have a milky white person, very dark black, and a bunch of variations where the skin color becomes increasingly darker. So you recognize polygenic inheritance because you have a continuous variation. You don't have A or B, or not even A, B, or C, but you just have a bunch of them with very little difference between each one together. There's a big difference between the beginning and the end, but every transition is just very gradual in there. That's polygenic inheritance. In the case of the eyes, I haven't checked lately, but the last time I checked, there was 9 genes that were working together just for your eye color. Now, obviously, we said that the traits are forms of genes and the genes are located in chromosomes, but when it gets really interesting is when some traits are on sex chromosomes. Because now when you're looking at genetics, it's not only whether you get that trait or not, whether it's dominant or recessive, but you're also going to consider whether the offspring is male or female. And that's what happened with my granddaddy's bull. Now, the X chromosome is big, so it can have over 1,000 genes. So the genes that are found only on the X chromosome, we call them X-linked genes. The Y chromosome is very small, contains about 80 genes, and genes present only, sorry, I need to fix that on the Y gene, there. I don't know what I was doing, but they're Y-linked genes. Okay, that's what I put in the ad. I need to remove this in here. Males cannot be, whenever it's an X-linked gene, is a male homozygous or heterozygous. And you say, wait, can't be either one, because homozygous means 2 identical traits. Heterozygous means 2 different traits. But we just said that there's only 1 X chromosome in the male. So there's a term for that. We call it hemizygous. They have only 1 allele for X-linked. And that's going to be very important because they can pass the allele to their daughters, but they don't pass it to the sons because they pass the Y chromosome to the sons. So X-linked recessive alleles are always expressed in males. Why? I just said it. The male can not pass the Y chromosome to the females, so the female is going to get 2 X chromosomes, one from the father, one from the mother. They may be identical. We're going to you could have homozygous dominant, you could have heterozygous, you could have homozygous recessive, but in the case of the male, there's only 1 gene. So that's why those X-linked recessive alleles are always expressed in males. Okay? So, like I said, the X-linked recessive conditions are passed from the mother to the son. The mother is the one that provides the X chromosome from the son in there. And typical examples are hemophilia or color blindness. I know this video is going to get too long, but I was 15 or 16. Spain was still a dictatorship, but there was a crown prince that was supposed to eventually take over, and someone decided the crown prince was 30 years old, he had to get married. Okay. We are going to have a selection among all the princesses of Europe to find a proper mate. And I got so excited. This is a Cinderella, right? And I could imagine balls. I could imagine, well, they're not going to have carriages, they'll probably come in limos, but they're going to have all of these balls in the royal palace, and there's going to be beautiful princesses in beautiful gowns and all of that. I even went to the extent of going to a royal palace and looking at the trees, and there was a particular tree that was really tall, and I said, if I climb on this tree, and I had to climb probably hours before this is going to begin, but I can look into the ballroom. I was so excited. I couldn't control myself. Remember, I was maybe 15 years old, okay? So romantic, right? You know what they did? Well, obviously they contacted only the single princesses, and they asked them to submit themselves to genetic testing. because hemophilia is rampant in the European royal families because of inbreeding. The prince did not have hemophilia, and because that's an X-linked trait, they knew he didn't have the gene. They check all the princesses. If you're a carrier, because you could have hemophilia, let's say that you have the normal gene and the hemophilia gene, you're normal, your phenotype is normal, but you're a carrier. So you could have a princess that seems apparently normal, but will carry the gene, and some of the offspring, some of the male offspring, will have hemophilia. So to avoid that, they did genetic testing, and there was only one princess that was single and was not a carrier in all of Europe. It's kind of like, this is not very romantic, genetic testing. So he ended up marrying the princess of Greece. To further ruin my dream, she was as ugly as a bulldog, and she was a very, very mean person too. But you can see how you can have a healthy male. The daughters, they're going to get a healthy gene from the father, and they can get either a healthy gene from the mother or the sick gene. The daughters are not going to get hemophilia when the father is healthy, because all the daughters are going to be big H, or big H, little h. This one is a carrier, but she's healthy. On the other hand, I hate when it does that. Excuse me. Sometimes the pen does that, okay? But when they talk about the father, and okay, so I'm going to have to write someplace else. It's pretty obvious. It's not going to let me do what I'm doing. When we're talking about the sons, they're going to get a healthy, depending on whether the mother's healthy or not, it's going to have a healthy X or the hemophilia, and then the Y. So we said the daughters, half are going to be healthy, half are going to be carriers. The sons, half are healthy, half have hemophilia. So you understand what was so important. They knew the prince was healthy. They didn't want to marry a carrier because they didn't want hemophilia to continue in the family. And that's caused, like I said, because the males have only one X chromosome, comes from the mom. So X-linked recessive conditions are passed from the mothers to the sons, and that's a relatively common condition. Look at Drosophila, the fruit flies. You have red eyes and white eyes. They are located on the X chromosome. The red eye is dominant. So when we're doing a Punnett square, not only we want to know how many of the offspring are going to have red eyes or white eyes, but we want to know if they're males or females. So the male, let me write bigger because I don't think you can see why the female has white eyes. So because that's recessive, has to be homozygous recessive. So what are the gametes from the female? It can have the little w, the other one has little w. The male can have the X with the big W or Y. So when you cross, you have X big W, little w, that's a female because it has two X chromosomes and has red eyes. Or you get this one, this is the big W, this is little w, so you have another female with red eyes. What one is the Y? Well, you have the little w and Y. It's a male because of the Y. It has white eyes because it has the little w, and the same thing with the other one. So notice in here, because that's what makes it very interesting, you cross a red eye male with a white-eyed female. And in the offspring, all of the females have red eyes, opposite from the mother, all of the males have white eyes, opposite from the father. And there's all sorts of combinations with fruit flies. One of the most fun things I remember from genetics lab, crossing fruit flies. But the point I'm trying to make is that when you're making a Punnett square, you not only want to say how many red eyes and white eyes, because you're going to say you have 2 red eyes and 2 white eyes, but you have to indicate the gender, male or female. Okay, so you have to be very careful with the answers, explaining are the sons, daughters, and what particular trait they have. That's the only difference, but again, I have a worksheet with some prompts that you can do to practice. Linked genes, you know that every chromosome has hundreds of thousands of genes, depends on the size. Some chromosomes are bigger than others. So when genes are physically located in the same chromosome, we say that they're linked. But remember, crossing over, also called recombination, can disrupt that process. We did that, remember, meiosis, and all of a sudden, yes, these genes that were linked, the big A and the big C, now you have a big A and a little c. So these links during recombination, they're not linked anymore. They're on separate chromosomes in here. How likely is that to happen? It makes sense that the farther apart the genes are, the more likely it is to happen, because there's more room in there to combine. Between A and B, to separate A and C, you can break this chromosome in many different places. Between A and B, you have less places, so it's less likely. And actually, people have placed genes in a particular order on a chromosome based on the frequency of recombination, saying, okay, if it's very unlikely to recombine, they're going to be very close. If they have a high frequency, they're going to be farther apart. And actually, even the units refer to number of recombinations. So that's a good way to locate genes on a chromosome. We talk about just now the X-link and Y-link genes. Link means they're located on a particular chromosome, but that can be disrupted, like I said, through recombination. That doesn't happen with the sex chromosomes, at least no between the X and the Y, because obviously the Y is too small for that. The last term they're going to learn is epistasis. That means interaction between genes so that one gene masks or interferes with the expression of another gene. That seems kind of complicated. But look at mice. They have 2 genes. You have the gene C and the gene A. C is going to mask A because when the big C is present, you do have a coat color. Basically, if you have little c, there is no coat color. You have a white mouse. Okay, let me just put in here white. But then if you look at A, it can be black or brown because one is dominant, one is recessive. So you're combining 2 genes, you can go through the Punnett square in there, but notice that when you have a big C, all you know is that they're going to have color, whereas black or brown depends on whether you have big A or little a. When you have 2 little C's, you get white mice. So you get 3 different colors, not because you have 3 alleles or 3 genes, but because of epistasis, because gene C masks. When you have little c, basically what they're saying is you don't produce melanin, so your fur is not going to have color. It doesn't matter whether you have big A or little a. You don't have the physical means of producing melanin, so your fur doesn't have any color. End of the story. That's one gene masking the expression of another gene, and that's epistasis. Once again, practice makes perfect. Do you want to do examples of genetics? Do you want to meet with me one-on-one? The video description has a link to my Wycent account. I'd be glad to meet with you. I'd be glad to share my problems with you so you can practice.